Good morning, everybody. Can everyone hear us all right?
Yeah.
Great. Look, welcome to the eDNA session. Thank you for coming. This is a bit of an experimental session for us. We've not run anything like this before. We should introduce ourselves. I'm Cris. I'm a professor of molecular medicine in the university, mainly based up at the medical school, but I do a little bit of work down here. And part of my role is being very deep into DNA technologies. And my name is Thierry Lints, and I work with Cris at Fortune. And my role is really to sort of be involved in the public interface with respect to genomics and medicine. So I do quite a bit of outreach work, and we'll see some of that today. And that's also partly why we're here as well, so that the public can come to grips with genomics. And this is a session very much driven by Rosemary and Scott as part of the Auckland Museum Institute, which is the Auckland branch of the Royal Society of New Zealand. They are part of it. So thank you very much, Rosemary and Scott, especially for making all of this possible.
So first of all, I was going to give you a little bit of a background. So obviously, environmental DNA is shortened to eDNA. And the thing about it is it's one of these technologies that's becoming increasingly accessible. There was a lot of hype about it a few years ago, but it's actually got to the point now where it is a genuine citizen science activity. People around the world who are not scientists by training are genuinely doing eDNA sequencing to better understand environments. It isn't mainstream yet, but we believe it's going to be mainstream within the next 3 years, which is why we think it's a really good time to start to talk about it much more widely. The idea of eDNA is that you get the DNA in samples out of soil or sediment or water columns, whatever you want really, sometimes even air, and you can sequence that DNA. There's DNA everywhere because there's organisms everywhere, bacteria, bacterial spores, viruses, as well as much larger beasts like us. We had a project a couple of years ago where we were going to actually sequence the DNA in the air in Parliament above each side of the debating chamber, but it got pulled at the last minute by security people. I don't think they wanted to see what was in all the hot air. But you can also use eDNA to look at the human impacts of the environment, of course, and environmental impacts on humans, which is where I'm interested in it. I'm a medical doctor originally, and I'm very interested in the interface between environment and humans. But from my point of view, what affects the humans and how our destruction of our environment may well destroy us. So the science of doing the sequencing of DNA is called genomics, and then the science of understanding what all of that massive DNA sequence data is called bioinformatics. And the idea of bioinformatics is that we've got a tsunami of data now. So when I first did statistics at university, we had these tiny little bits of data that we could visualize in an Excel spreadsheet. And I regularly analyzed terabytes of data. We just downloaded 2.5 petabytes of data from overseas that we're analyzing. You try and open that in an Excel spreadsheet. So the idea of bioinformatics is often these days dealing with very big data. So what we hope you're going to take away today is a hands-on introduction to the technology of eDNA. We're not going to dive deeply into the biological interpretation. We just can't do that in one day. This is around the technology, so you understand the technology so it doesn't seem foreign, and so you can judge yourselves whether it is useful or not for your specific work in the future. You'll have a chance to understand DNA sequencing. You can have a look at DNA sequences. We're going to go through some data. There's some little piece of paper with URLs on the table that some of you have already picked up, and that links to a download that is a safe download. It has a copy of an old, slightly older version of this presentation and some data that you're going to have a play with if you want later on in the session. We recognise that eDNA is not the be-all and end-all. You guys have got masses of tools that have worked really well for you. We're not saying eDNA is going to supersede any of those tools, but we're suggesting that perhaps in the future this could be another really helpful tool in your conservation toolbox or environmental science toolbox or whatever your area is. And I should say that Thierry and I are not environmental scientists or biodiversity scientists or phylogeneticists. We both work on the human side, but we are experts in technology. And we can refer you on, though, to the right environmental people, statistical people, if you have a particular interest. So very quickly going to run through just to introduce you to some of the very basic science around this. And please don't be insulted if this is very much old hat to you. But obviously the idea that all living organisms have DNA, and this is used as kind of like a plan or a template for building proteins that form the basis of the structure and function of every cell. And this DNA is transcribed into RNA, which is kind of like an intermediate map, which is then used as the template to make proteins. And the DNA is obviously arranged into genes, where each gene in general encodes a protein. And the sequence of the DNA bases is the sequence of the protein, or a plan for the sequence of the protein. And every different species obviously has parts of their own genome or their own DNA that are quite distinct. But quite a few species are homologous, or they're closely related in evolution, and the parts of their DNA can be very similar, with only a few bits distinct. And even obviously within a species, like within humans, we all share 99.9% or more similarity. And that means that if you look at the sequence of our DNA, many of us are really similar. And there was quite an interesting event happened for the first 3 humans who had whole DNA sequencing. There was a Korean and 2 Caucasians. And one of the Caucasians and the Korean were much more similar in their DNA than the other Caucasian was. So that kind of shows just how similar we are across races and ethnicities, doesn't it? Okay, so the idea of eDNA is you're looking at an ecosystem and you're not sequencing a single individual or a single species. You're getting all of the DNA from all of the cells in an ecosystem. You're pushing it all together and getting DNA reads, which are these little reads of maybe a few hundred bases or individual building blocks of DNA. And these reads will come from any different species in your ecosystem. And if there's one species that dominates your ecosystem, they'll contribute many more reads than the other species. And because parts of the DNA of each species are distinct, you can take these reads and calculate the proportion of reads in your ecosystem that comes from all of the different species. And as you'll see when you start playing with this data later, there's a lot of different species, even in a water sample. And there's a lot of applications for this. We can use it for species identification. There's been some really cool things where people have used small DNA sequences, prototypes of them, and up in the mountains, they've seen a plant they didn't know what it was. And as a demonstration of the power of the technology, they've mashed up the plant, extracted DNA, and run some DNA sequence and identified it based on its DNA. It's not mainstream, and when I go tramping, I don't have a little DNA sequence in my pocket just yet, but that's not that far away. And the costs aren't that great. So that will become a thing before too long. But obviously monitoring species at risk, monitoring invasive species and trying to identify invasive species. For example, monitoring water in streams. Is there any evidence of an invasive plant that has got into the water upstream? The idea of keystone species. These are species that can indicate a much larger piece of information about a water column, or a stream, or a terrestrial environment. And the proportion of that keystone species in the ecosystem can help you identify pollution, human fecal contamination, and so on. And the idea, of course, of monitoring environmental remediation over time. Obviously, in human health, there's a lot of implications. I'm Deputy Chair of ESR, which is a Crown Institute that does a lot of the science behind the COVID response. And as part of that, as you may have read in the media, ESR is doing DNA sequencing of sewage and also of outfalls of various sewage outlets into streams and so on to try and identify where is COVID-19 in the environment. Are there particular areas of Auckland where there's a COVID-19 infection that hasn't been diagnosed, but we can see it in the water, then we can look in a public health response much more closely. So a range of applications for it. Obviously, forensics is a big one. We can now track people around the place experimentally by the little bits of DNA they leave when they hold on to a handle in a train, for example. And it's a little bit scary, that level of eDNA, but it is becoming possible. There's been some really fun exercises where people have been tracked around New York, for example, by swabbing DNA off train doors and car doors and building doors and so on. So a lot of different applications. So the bioinformatics we do works by taking all of life, all of the DNA across all of the different phyla and kingdom, and breaking the DNA sequences up into these tiny little pieces, often around 20 of the DNA bases long. So you have these sequences of 20 A's, G's, C's or T's, the 4 letters that build up DNA. And then you take your sample, where you've got lots of different reads of sequence of a few hundred of these pieces long from your environmental sample, and you break those up into little tiny pieces of about 20 bases. And then you just try and match them all together, a simple matching game, like playing a matching game with cards. And we do it like this because computationally that's much more tractable than taking a long genome, for example, of a human, 3 billion base pairs long, and then taking lots of individual DNA sequence reads from the environment and trying to match it to the human genome. That's a really hard thing to do, but it's much easier if we break them up.
Sorry, I'm not sure if we're allowed to ask questions.
Of course you are.
Just the question about if you're taking a segment, how do you know which segment to take and work on?
So in terms of a known genome of an organism that you're using as kind of a reference to map to, what we do is we take 21 base pair pieces that we call k-mers, and we take the first 21, and then from position 2 to 22, position 3 to 23, position 4 to 24, all the way through the genome. And when we have an environmental sample that may be, say, 100 base pair long read, we do the same to that. And then we just do a matching exercise with all those individual pieces from the environmental sample to all the pieces from the genome. So we're not selecting any one piece. Actually, what we really do in practice is a little bit more nuanced because there are some pieces that are much more common in some genomes because they don't get degraded in the environment. So we actually would focus on them first sometimes.
Thanks.
In addition to the sequencing devices that we'll explore with shortly, there's been miniaturization and sort of optimization across all the different steps post-collection to be able to extract DNA. So it's now sort of more or less a kit form process, and I'm just going to sort of go briefly through the sorts of steps that we've taken in the Science Outreach project that is going to be described shortly. In that project, we were working with some people from Kim Handley's lab at the School of Biological Sciences, their groundwater team, and we were sampling out in the Oamaru Awa in Tamaki, Glen Innes area, with year 7, 8 primary school students. And the first step was to filter some of the water at different points along the Awa, and we'll come to that in a little bit. And from that, we largely collected microbial material on some filters But in addition, in the mix, there was also some eDNA that had been released and was in the water in sort of a non-organismal form. So the first steps you can sort of see are to disrupt the bacteria. And so that's just done by using a vortex mixer that shakes things up very rapidly within a small little tube with some silicate beads, and that sort of cracks open the bacteria. And then that material was then transferred onto into a soil optimized DNA extraction kit, which is all done in very small tubes, very conveniently, through a number of different steps of centrifugation and purification over a little column, which selectively binds the DNA, and then you can wash and dilute that DNA, and now you have relatively pure DNA that you can process for the rest of your experiment. And then that is then transferred into a workflow which prepares the DNA for the actual sequencing process using these particular devices that we have here. And so the steps are so that when the DNA is prepared through this bacterial breaking up and then spinning through columns, etc., it's sheared, so it's broken up into pieces. You can optimize for very small pieces, or you can optimize for very large pieces. We didn't pay too much attention to that, but those pieces have sort of broken ends, so they're not very tidy. And so the first step is to repair the DNA ends with an enzyme. And then after that, little barcodes, which are just little short stretches of DNA that are sort of diagnostic for the particular sample, are added to the end. So what we did in our sequencing was we pulled multiple samples together from different sites, and we wanted to, in the bioinformatics step, to be able to identify which sequences came from which site. So they get a little tag of DNA sequence, which allows us to make that determination. And then after the ligate barcode step, there's another cleanup of the DNA, and then some adapters are placed onto the DNA sequence. And those adapters allow the DNA to actually interact with a protein pore that's in the membrane of these little flow cells. So you'll be able to have a look at these tiny little flow cells, which really are the guts of the sequencing device. There's the electronics, of course, but in the little window that you see here, in these flow cells, there's embedded about 2,048 little protein pores in an electrically resistant membrane. And as DNA is threaded through those pores, it changes the current across that membrane, and then that is registered and then sort of deconvoluted to be able to figure out, what's the DNA sequence that's going through that pore at any one point in time. So just interrupt if you want to ask any questions, of course. So this is the device, no point in belaboring that since you've got one in front of you that you can have a look at. And this is not the limit of miniaturization that these have been driven to. The same company now produces a small device that can fit onto and interact with your cell phone, as shown here. These have been something that have been in the wings for a year or more.
2016.
2016. So this particular company has a very sort of aspirational production timeline, where you're sort of interested in waiting for the next cool thing to come along. It takes a little while for them to actually see the light of day. But this will,
Sorry, I was just wondering, I was interested in how accessible it is. When I was helping Thierry set up the lab, he told me that unit cost $10,000 for the actual unit. But bear in mind that these machines used to be huge and tens of thousands. I mean, how much, if you wanted to set up yourself and actually do some eDNA, what's the minimum I'd have to pay without getting hugely sophisticated?
well, I mean, I think for a start, if you have one of these devices, and of course the sequencing costs are coming down all the time, the smidge iron device probably doesn't cost a whole lot.
I mean, usually the particular company we're working with called Oxford Nanopore basically gives us stuff for free because at this point they want to build the technology. technology. Their business model isn't, if I understand correctly, is not to make money with devices, it's to make money with the reagents.
Yeah, it's sort of like a computer printer business model where you pay $100 for a computer printer and then you have to pay hundreds and hundreds of dollars every year for ink. So it's not dissimilar from that.
It's not quite as bad. We'll talk a minute about a number of other citizen science projects that have used these technologies. And most of the people in those projects have not paid very much. And obviously the business model will mean that they pay eventually. But we don't think it's a volume model. It's not a high gain model on any one device. And the idea for companies are to get everyone having these. So they cost probably, a few $100 with a few $100 reagents each year. I think it's the aim.
Yeah, and also to Rosemary's question, I think it also very much depends on the particular need that you have. And so these are actually a sort of a larger form of the flow cells, and I think they typically run around about $800 or so. But there are smaller forms of flow cells that have less throughput, which are only about $100. These are called flongals, and they have a little bit less capacity in terms of the amount of DNA that you're sequencing throughput, but they may be sufficient for some needs perhaps.
And you'll see when you start looking at the data that we've got way too much data, even in this little exercise we've done. So a smaller capacity one would have been just fine.
Okay, so this is really just to sort of highlight the different sort of levels of organisation across the animal and plant kingdoms, or across the domains of life, sorry. And so you have sort of taxonomic organisations where the taxa are organised into different characteristics based on their characteristics, and clades, which are based on their sort of common hypothetical ancestors. And then we have comparative genomics, which is really sort of in many cases really challenged formal earlier taxonomic thoughts about the sort of affinities of different organisms. In terms of trying to understand all the domains of life, there are now some really massive programs that are going on around overseas. So there's, in the British Isles, there's the Darwin Tree of Life program, which is set out to sequence all 60,000 known eukaryote species in the British Isles in the next decade. And then there's the Earth Biogenome Project, which is aiming to sequence the genomes of all 1.5 million known eukaryotes in the next decade. So the phenomenal, that's a real, as Cris said, it's a tide of a tsunami of data that's coming along as a consequence of this. Just try and play that.
As many of you will know, there's large Australasian biodiversity projects, and some people within the Royal Society of New Zealand and University of Auckland and AUT are involved in these. The particular project that I've got up here is the Decadal plan for biosystematics. I think actually some of you in this room may have been part of this. One of the, if you read through that plan, one of the things that comes out in it is the idea of genomic methods and how they provide unique opportunities. But the particular point they highlight is dark life or dark DNA. The idea that when you start doing DNA sequencing, you find all of these new organisms, or at least new DNA sequences that you didn't know about before. And traditional phylogenetics and traditional science of taxonomy may have missed these organisms, but they may be very important components of ecosystems. So this dark matter, biodiversity dark matter, is a whole new science that's coming out of this. And there are large projects in New Zealand and Australia that are leading this.
Okay.
Thank you. Super interesting. I really appreciate it.
We do it all based in Cali,
Yes.
so I wondered whether we could.
Those are the leads, redents, and then there's sort of various QC scores and things like that. These are the number of different barcodes that have been read for the 5 different samples. So you can sort of see that there's, just in this particular run, while we've been sitting here, there's been 30,000 different DNA sequences read that had barcode 1, 40,000 that had barcode 2. So we've read while we've just been here, a couple of 100,000 DNA sequences just while we've been here. It's amazing to me.
How much power does that thing use, and can you drive it with batteries in the field?
Yeah, I think so. It's been in the links that we've sent. There's like some field work that they're showing of people doing this in sort of Kenya and stuff like that. So yeah, it's been sort of designed around the idea of portability into the environment.
And this thing slots into there.
Yeah, so basically that this is the other version in which you would just plug this into a laptop. So this is called a MinIron. That's a slightly different thing, but but yeah, so they just plug into here. These are the pins that sort of interact with the device and goes into there. There's a little sort of pad here that provides set temperature across the series, and then just slot it in.
They've only made it
like so.
Okay.
And then just plugs into your laptop.
Wow. Can I take a photo?
Yeah,
please?
sure. Go ahead.
And
You said to me, well, it's not that far off in the field.
the
You spit on your phone.
preparation of the samples.
Yep.
Please go see your doctor.
No, so I mean, again, they're doing these sorts of things out in the field.
I do a little bit of
Preparation for the DNA prep took about a morning, and preparation for the taking the DNA and putting on the adapters ready for sequencing. That took an afternoon. So within a day, you can go from sample through to loading this, and then letting it run overnight. Or normally, if you were trying to get as much sequence from it as possible.
But as Cris said, the ethics of it all, right? If you've seen
But,
BlackBerry, you can just imagine, you go into a concert and it goes, no, sorry, your core temperature is too high. We think you may have a virus. You're not allowed into the stadium today.
Yes.
Wow.
See you later.
Thank you very much.
Thank you again. Do get in touch next year if you
Are people doing population work with eDNA yet? Or is it
Probably have to scrap most of this audio.
There's also citizen science projects using these technologies now. There's on the left here is a project to address trafficking in endangered species, where sometimes the only evidence of, for example, a rhino horn or an elephant tusk is actually the rhino or elephant DNA that may be left in traces in packaging and other materials. And there are citizen science projects in Africa sponsored by the Smithsonian Institute to try and use eDNA as a way to get evidence to prosecute. On the right-hand side is a really cool project in Cambridge in the UK where I worked for many years. It's called PubSci. It's a citizen science project to use exactly the same machines that we have here to, over time, monitor the quality of the River Cam and some of its tributaries and associated ponds and so on. So hopefully in this first part, we've given you a very basic scientific background and a basic appreciation of the context in which this area of science is developing. A lot of the science involves very high-end genomics and bioinformatics, but it is now becoming very accessible to any of us, and that's why we think it's so exciting. I'm now going to hand over to Thierry, who's going to talk about a sampling project that we've done that's generated some data that you're going to look at.
Okay, so as I alluded to before, this is a schools project that's out in the Tamaki Glenlinnis area with a number of sort of contributing schools that are involved. And for a few decades now, one of the main sort of waterways, the Oamaru Awa, which is shown in the long blue line there, has been sort of a source of concern for the community because of pollution in it. And so one of the things that we wanted to set out to do was get some information about the microbial content within the awa. And in addition, we took another couple of samples due to the proximity of different schools with different areas. So we went also into a place called Johnson Reserve here and collected some water from a little pond that had some eels in. And Point England School also has an eel breeding program going on with some tanks, and so we also sampled water from those tanks. And then on the left here, we have Emily Gios and Olivia Mosley, who were from Kim Hanley's groundwater team, who are helping us collect the samples. And this is just some other students from a different school interacting with the team. And let's see what okay, so going through the sort of the protocol that I showed you before in terms of how the DNA is extracted and then prepared to go on to the anion with Annabelle's help, we sort of loaded one of these flow cells, which you can come along and have a look at and inspect. Basically, it sort of works on a capillary basis to bring the DNA sample that you load ultimately through a small little port here. And that DNA sample just sort of gently floods across this membrane. And as I said, the membrane has in it 2,000 proteinaceous pores, and each one of those pores is hooked up to the ASIC that allows the changes in current through that pore to be registered. And then a neural network sort of algorithm sort of identifies what the sequence of bases is based on changes in the current across that particular pore from moment to moment. So as DNA streams through the pore at around about 400 nucleotides a second, you're getting this sort of change in electrical current. Now, one of the great advantages of this is not one of the other major methods in terms of DNA sequencing is called sequencing by synthesis, for example. But this is just using a DNA that's not amplified being fed straight through the pores. And because of the way that this operates, it's able to produce very, very long reads of DNA. So we didn't pay particular attention to the read length, or the length of DNA we were aiming for. But as you'll be able to see when you look at the device and the output that it shows, some of the DNA fragments that we're sequencing are of the region of about 20,000 to 30,000 base pairs long. The sort of state of the art is to be able to sequence something of the order of about 2 million base pairs in one read, one sequence of 2 million base pairs of DNA going through the pore at one point in time. So that's where we got to that. It might be a good time to have a look here. And
I'm trying to get the camera on it, but I can't, unfortunately. The system doesn't seem to. So people want to come up. Maybe some come up now, and some come up during the period of the time. Oh, you cool. Well done. All right. Well done.
Oh. Okay. Brilliant. All right, so it was good timing. Okay, so what we have here with these little colours that are flickering on and off, that's the representation of the 2048 pores that are in the membrane. And of these, because we've been running this for now 3 or 4 days off and on, as time goes by, you start to see pores dropping off, so they're not being active any longer. But the ones that are green, bright green, are actively sequencing. As I said, and it's a bit hard to tell, there's actually supposed to be two different colors of green, so there are pores that are sort of not actively sequencing, but are ready to receive DNA. And then there are pores that have just sequenced and are now recovering before before they're able to receive another strand of DNA, and then there's a whole bunch of pores that are currently inactive.
Could I just say something about the pores? Because this is nanotechnology in practice. These pores are actually from bacteria. There's a range of different bacterial proteins, and these are actually called hemolysins. Bacteria like Staphylococcus bacteria use these as little pores that they transfer material into other cells with. And what has been done here is these pores have been taken out of the bacteria, recombinantly engineered into membranes, which we're then sequencing through. And as each molecule of DNA from the environment squeezes through the pore, there's a voltage across a little membrane in which the pore sits. And that voltage changes a little bit as different bases, A, G, C, and T, squeeze through the pore. And very clever statistics untangles the different pores and the voltage changes of the different pores as all the DNA is running through them. So I think this is really cool because it's a synthesis of bacteriology and maths and nanotechnology engineering. All of these different things had to come together to put this into a apparatus that can be used.
Yeah, and of course, if you're dinosaurs like myself and a slightly less dinosaur like Cris, the idea that you can sequence 400 base pairs in a second is just absolutely phenomenal. I mean, it took me about a year to sequence a couple of thousand base pairs when I was doing my PhD.
Tell us.
Sometime ago.
There's
No, I'm not going to say how old I am.
Does the hardware wear up? Like, I presume it has a maximum duration of use.
I think these are all. I don't think we've encountered any particular obsolescence in terms of hardware yet. What does change more frequently is the particular flow cells and the proteins that they have embedded within the membrane as they continue to optimize, making sort of mutational changes in those protein pores to work better for particular characteristics. One other approach that's very valuable with these is, as I said, this is not sequencing by synthesis, so that you're just using native DNA or native RNA to sequence directly. So DNA and RNA is often modified in organisms in important ways, and so this is one of the few platforms where you can really understand what the nature of those modifications are, because they have very important consequences in terms of the regulation of genes. And the fact that you can sequence directly RNA allows you to sort of get a window on how an organism is responding to the environment in terms of turning its genes on and off without the biases that normally come along with actually having to purify the RNA and then turn it into DNA and then to sequence it. So there's a lot of sort of really powerful aspects to the use of these devices, these flow cells.
You would have heard Mike Bunce in the video talk about barcoding, and what he was meaning is to make it. Oh, great. Well, what Mike was meaning was that in order to make it easier, there are particular sequences across all of life that vary more than other sequences. And for example, to look at bacterial communities, the 16S ribosomal genes have particular sequences that vary in a very characteristic way. So you can limit your sequencing to just them in order to identify species. For microbial eukaryotes, the 18S ribosomes, and then different sequences for plants and animals. What we've done here, though, is we've made it much easier because the technology is now getting better and better. We've just sequenced everything. Every bit of nucleic acid or DNA from the environment we've sequenced. And the point of that is that it makes it a heck of a lot easier without having to do special extra steps to just in the field, without being a scientist, to just get the data. So what I'll do now is just have a look at the data.
I'll just toggle through here for a moment, Cris.
Oh, yeah.
Just to show. So there's a number of sort of graphic displays that this little device has. But this is just an indication of how long some of the DNA sequences are that we'll be getting on this in a very non-optimal situation, I guess. But as you can see, some of those sequences are as long as sort of peters off around 20 kilobases, 20,000 base pairs in length. But there are sort of a few reads that are quite a bit longer than that. But that was without any sort of effort to try and specifically look for purifying long intact pieces of DNA.
And that is, in some cases, as long as the genome of the organism. And some of these genomes are actually circular, and very occasionally you find you read all the way around and then you start to go around again.
One of the issues with this type of sequencing is it does have some errors, and it's a trade-off with the technology. And if you ever think about it, if you think of your most basic statistics, one way to overcome random error is to do something many times, because the random errors won't tend to stack up if they're not systematic. And that's one of the ways we understand sequencing much better. We read the same bit of DNA many times, so the errors kind of cancel one another out. I feel embarrassed explaining this in front of a professor of statistics. We'll pass over that.
Well, I was wondering about that. Obviously, it's good not having to know the finer details and everything, but is there a risk of contamination of, in the process of breaking it down? That was a thought in my mind. Could you get false positives or false readings?
Yeah, yes, you can. And for example, you can get false positives from your hands. And there's been various forensic cases where it's turned out that the DNA that was thought to be from a criminal was actually one of the lab staff processing the DNA. And we even find this sort of thing in some of the work that we're doing in human health, where every now and again we think we've got a very important mutation in a cancer, but it turns out to be a contamination.
Yeah.
And when you run things through a sequencing machine, there's always a little bit of DNA that hangs around from the previous run. Big commercial sequencing companies will sequence all sorts of different species. And if I look very carefully in human DNA sequence that I've had sequenced in a commercial company overseas, I'll find eels and cows and all sorts of things, very, very low levels, but they'll be there. So we have all sorts of statistical ways of dealing with the contamination and getting rid of it. Okay, so those of you who have computers, I encourage you to spend a few minutes exploring the data yourselves. And if you don't have a computer, if you just get on a table with someone who does in a few minutes when we go through it. I've got here in the presentation that you may have been able to download with some of the data, a few links. And what I'll do is I'll go through on the screens a few ideas for you to have a think about before you have to go through, if that's all right. So one of the first things I'd suggest is just looking at the idea of particularly important organisms. So the first one I've got up there is you may be interested in looking for E. coli, Escherichia coli. This is a organism that's a commensal that lives in the gut of many mammals, and looking for this in environmental samples is a really good indication that there may be some human fecal contamination in that sample. On the second line are more species-specific evidence of contamination from feces. Streptococcus faecalis, bovis, equinus, and avium. You can work out from the names the different species they are indicators of. Just for fun, there's thermophiles and acidophiles. These are bacteria that are specialised in surviving in really extreme environments. And if you have a look, you may find one or two of them here. Some of them may be there just simply due to error, sequencing error, like you were talking about, but others will genuinely be there. And it just indicates the breadth of life and that we actually do find things everywhere. You'll also find Staphylococcus aureus. And I said, why is that of interest? But unfortunately, I gave the clue away before by mistake. Staph aureus is of interest because it's kind of a circular thing. The Staph aureus is the bug that we got the pores from that are in here. So it's quite cool that we're sequencing Staph aureus by squeezing its DNA through the same pores that the bacteria itself have evolved much better than we could ever engineer. So what I'm going to do first is just go to the internet. I'm just going to change over to a different screen. So there are a number of files in your hard drive, you would have downloaded that end in HTML. These are samples that have been processed through our bioinformatic pipeline. It's very easy to do. And the bioinformatic pipelines end up with producing an HTML file, which you can alter and play with. This is a system called Krona. And the idea of Krona is that you're representing everything as sectors of a circle. So root at the center of the circle, this is all of the DNA sequences of all of the different organisms, taxa, that you can find. If you then go out to bacteria, bacteria is probably 90% of the circle going around. There's little bits in these blue and green colours that are not bacteria, but everything outside of this red circle here in the bacteria is different clades of bacteria, gradually going finer and finer detail in the clade in the tree of life. So if you look in the bacteria, there's the proteobacteria. These are bacteria, I'm generalising hugely here, but these tend to be gram-negative bacteria, the bacteria that are often adapted very well for wet environments. And then there's another phylum of bacteria, the bacteria here that I've got, trying to, sorry, my computer's just frozen. Here we go, the Terrabacteria. These are bacteria that have adapted for terrestrial environments in general. In general, they're gram-positive, they have these thick cell walls that may be part of that adaptation. And then as you come around further, you've got other groups, the FBC group, the PVC group, spirochetes, and other types of bacteria. And let's just say if we go to the Terrabacteria, if you click on that, it further expands, so the whole circle is just those Terrabacteria. And let's say we were just most interested in the Actinomyces bacteria. If you click on that again, and so on. And you can see this huge range of different bacteria. You can also do a search. So if you were interested in, let's say, Clostridium, you can type that into the top corner, and you need to spell it right though. And this will start to, if I go back out, sorry, I should say you can zoom in and out by just clicking on the right-hand side or the left-hand side. If you search, it'll show you where they are. So they're over here. And if I click on this, you can then expand it out more, and you'll keep on seeing where these Clostridium are. And you can keep on expanding. So this is a range of different Clostridium species which have appeared in the DNA data. To go back out, you can either click inside, or you can go over to the right-hand side and click at these various levels. So I'll go right back out to the root by just clicking on that. So you can have a play with that. I'm going to show you another thing you can have a play with called Parvian. And Parvian is another set of software that can load the files that come out of the DNA sequencing bioinformatic pipelines. And Parvian can show different illustrations from Krona that you've just seen. So with Parvian, you're going to have to load the files yourself, but it's very easy. The files are in that set of files you downloaded. They've got names like BCO1, BCO2. Each of these, there's 5 of them, and each of the files comes from a different sampling place.
Excuse me, Cris, is that in the Krona outputs file?
No, that's in the other one. Sorry, Rosemary. There should be another folder with another name.
Results file? Results
file, that's right. And if you go into results files, you'll see files named BCO1 through to BCO5. So if you go to that Parvian website,
it asks what you want to open it with. What do you recommend?
don't double click it. So if you did go to the Parvian website, instead of double clicking it, and the Parvian website was listed in the PowerPoint slide, and I can come around and help you get it, then just click on Data Selection and then Browse. Then use your computer to go to the site of the files, select all 5 of the files, and then just click Load. And it will progressively load onto the website all 5 of those files. And these will allow you to see things in a slightly different way. So if you click on Results Overview, they will give you an idea of how many sequence reads there are. So, for example, the BCO1 sample, which was taken, get where that was.
Sometimes it's on the Unruala.
On the Unruala, or the BCO4 sample, which was the eel tank, I think. They've got different numbers of reads. The BCO1 sample is 330,000 individual DNA sequence reads. And about 30% of them could be classified. That means about 2/3 of them, the software could work out what species it comes from. Maybe the read was a bit too short, or there were various reasons that it couldn't work it out. We don't want to make too many errors in this, so we're quite careful to exclude sequence reads that we're not sure of. And then there's about almost all of the classified reads are microbes. If you sequence a bit of water, there may well be mammalian DNA and plant DNA and fungal DNA in there, but it tends to make up a relatively small proportion of the diversity of life in that water. And actually, for this exercise, I've excluded plants and animals to make it a little bit easier to go through, but we can include those. So the cool thing about the targeting software, though, is you can start to see things in terms of comparing samples. We're also basically seeing a tree of life. So if I'll just go to the eel tank sample, and I'll come around the room and help people set this up themselves. But unfortunately, you can't quite see the whole thing here, but this is the sample arranged into the tree of life. You've got the bacteria here on the left. I'm just going to zoom out my window a bit so you can see it easier. So you've got the bacteria here on the left, and then you've got small numbers of archaea that I'll talk about in a minute, and then all the different bacterial clades all the way out. If you wanted to just focus on one bacteria or one particular thing, click on it. This is Clostridium Botulinum. And you can see that there's a lot more of this in sample number 4 than the other sample. Sample 4 is an eel tank sample. So you have a play with that. I'm just going to go back though and just quickly go back to the PowerPoint to give you a few thoughts about interpretation before you have a play with it. So the first is, in the PowerPoint file that many of you will have downloaded with all those sample files, one of the slides has a list of organisms that I regard as quite important in terms of being potential pathogens. And I bet you're going to find a whole heap of these in the water from these streams. Now, it doesn't mean that all the kids playing in the streams or doing our project are at risk, I hope. It's just a fact of life that these pathogens are often in our environment. And when we're doing monitoring of beaches or streams and health for public to be in them, it's a very nuanced process. It's to do with how much of them there is and to do with the form, are they in spores, can they be taken up and so on. So you may want to have a look for some of those pathogens. Let's just go through a few of them though. So you'll find Clostridium Botulinum there. And that may sound a bit scary. This is the bacteria that produces botulism. And it's a type of bacteria that's an obligate anaerobe, which means it can't live where there's any oxygen. So it has to live in an environment without oxygen. It's a bacteria that does cause terrible human disease, but not very common at all for you to catch it from a water source. It can very occasionally infect wounds where the bacteria gets into a wound and the wound seals over, there's no oxygen, so the bacteria can start to grow. But it more commonly produces a toxin that you can ingest, which gives you the disease of botulism. And you sometimes get that in canning processes. Many of you will know a lot about how to prevent this. Very occasionally it can be inhaled as spores leading to disease, but that's more in bioterrorism events where it has been released deliberately in terrible situations. But the toxin it produces is a neurotoxin. It blocks acetylcholine processing in your nerves. It's actually the most potent toxin known to man. It's also the toxin that's in Botox. This is Botox. And this is us harnessing this bacteria in industrial labs to produce Botox. And Botox isn't just used for cosmetic surgery, it's used for a whole
range of diseases where you've got muscles that are automatically contracting and cannot stop. And if you're interested, I've just got on the slide the New Zealand government, the MPI agency's website about this, which is really informative. So for any one of these different bacteria, there's all of these fascinating sides to them. The archaea, you'll see archaea there as a source of quite a few of these samples. Archaea are single-cell organisms that are very like traditional bacteria, but their genes and their genomes are much more like eukaryotes. So they used to be lumped in with bacteria, but many of you know far more about this than me. I'm almost embarrassed talking about it. But they're now separated out. And they're really interesting because some of them are very good at surviving in extreme environments. You'll find some puzzles in there. You'll find some, like the Helicobacter you'll find in the sample. They are normally only found in salt water, not in fresh water, but occasionally you can find them in fresh water. One of the really interesting things about them is that they can be used for carbon dating because of the way they fix carbon in marine sediments. Many of them produce energy from light, and they use bacteria rhodopsin molecules to do that. And those molecules are surprisingly similar to the molecules in our retinas that detect light. And this is an example of convergent evolution. We didn't evolve our light detection molecules from these bacteria. Instead, a completely independent solution was found in the evolution of mammals that evolved this almost identical molecules to detect light in us, as can be used to produce energy from light in the bacteria. Here's another really interesting one, the Eurovirus. So these are viruses, and they're actually not viruses that affect us. They infect bacteria. They're called bacteriophage. And these viruses have these incredible nanomachines in their tails that can puncture into bacteria to get into bacteria. And these viruses then hijack the bacterial's machinery to produce more virus DNA, more virus protein, package it up, and then the bacteria commit suicide under the instructions of the virus to let the virus out. The mycobacteria, you'll find lots of mycobacteria. These are the bacteria that cause a whole heap of human diseases. Leprosy, tuberculosis. They're in the ponds in Glen Innes, but they're not an immediate risk to anyone. We find them all around the environment. These are fascinating bacteria in that they've got very thick cell walls, and some of the disease-causing ones are obligate intracellular parasites. They can't actually survive for very long when they're free. And what they do is they get into human cells, especially immune cells, and they multiply and live in those immune cells. So finally, you're going to find some human reads when you have an explore. Why do you think this human stuff in the water? We're everywhere. We are everywhere. And the DNA that's in the water lasts for a few days probably before it gets degraded, so it's a fairly narrow time of sampling. But how would we get into the stream in Glen Innes?
After through sewage?
Through sewage, exactly. Sewage outfalls are a very big source of human DNA, and often human DNA and indicator organisms like E. coli that indicate the presence of human feces often correlate in the streams, not always.
As an aside, do you know if this is how Auckland Council gather that data as to which creatures are safe to swim in?
They tend to do it not through sequencing everything at this point, although there's several projects in the University of Auckland that have involved that, but they tend to more do it by very specific, much easier identifications of E. coli and counting the amount of E. coli.
Cool.
So the human DNA is really interesting, and actually I had a quick look through and I aligned human DNA to different genomes, and you can start to identify the mix of human DNA. In forensics, ESR won the Prime Minister's Science Prize last year through a product called StarMix, which is now used around the world to untangle mixtures of human DNA, just like we find here. I think that I can see at least 20 people's DNA in there, and with a little bit of effort, if I had access to ESR's police database of DNA, I wonder if I could go and see who those people were, but I never would. because think of all the ethical issues around that and all the privacy issues and so on. It's a fascinating area. So the science is starting to allow us, though, to do that, and that's something that we haven't discussed as a society. So you'll find human DNA in there. Have a think about how we could identify people, how it could be useful. So have a look, and we'll wander around. Just though finally here, we're not experts in this area. We're experts in the technology and in the human side, but there's a whole range of people we can put you in touch with, and some of them are in the room here. There's all of Kim Handley's team and other groups in the School of Biological Sciences in the university that have worked to put this together, and they're the real experts. There's groups in the School of the Environment who are great people to talk to about the slightly bigger picture. There's people in the Department of Statistics. There's ESR, if you're interested in the more practical side of environmental monitoring or forensics. And there's us in the School of Medical Sciences at the medical school, if you're interested in the human side. We're not experts, but we can come around now as you look at the data and we can talk to you about it. But do let us know if you want to get in touch with anyone. Have a look at the data for just a few minutes, and then we'll go through with a little bit of a question and answer for the whole group at the end.
Can you share me how to get into Darwin?
Certainly.
On your website.
Just keep hold of that link. The link will be open for 2 weeks, and you can keep downloading it again if you lose it. Have an explore yourself of the data. It's fun data to play with. The one thing it always surprises me about is the sheer diversity of life. The literally thousands of different organisms, even in this one little tiny data sample. And we've not even included plants and animals in there. So I guess I just wanted to end up by saying that there are pros and cons of these technologies, of course, and some of these technologies will be more difficult than you need. You don't need a fancy DNA sequencing technology always to achieve a goal. But I feel that these technologies are becoming so easy now that companies like Oxford Nanopore, who made this machine, are working with communities to try and get people using it more and more. And it's a chance to consider whether over the next 5 years this may become part of your own armoury, monitoring sequential health of streams, monitoring terrestrial environments, identifying where invasive species may have come into an area, identifying what is suddenly disappearing progressively from an environment, which is a particular worry. So if you're interested in future involvement, do come back to us. You'll have our email addresses, I think, and we're very happy to talk. We're happy to put you in touch with the real experts in the environmental science, but we're also quite well connected to different people using the technologies. In particular, if you wanted to get hold of some of these machines or technologies, the way we should do it is to coordinate as a community across New Zealand and go to the companies saying, look, we want to do this. This would be the advance to you. Give it to us for free. And it's much easier to do if you've got a large group doing it. So thank you very much for your time. Your next session starts in just a few minutes, but I thought just leave chance for any questions or comments or thoughts or things we can do.
Quick question. Soil samples versus water samples in terms of extracting DNA. Could you make a comment on that?
I know that you can do both. The kit we used is actually designed for soil samples.
Yeah, it's designed for soil samples, but I think what's been found from Cris and others is that it's because that's in some ways the most difficult sample, the seed sample, that it works well for whether it's water sample or fecal samples or what have you. So it seems to have the required inhibitors to allow the required parts of the kit that will allow you to red inhibitors fouling up your sequencing reaction. So it seems to be universally pretty useful.
Yeah, so soil very doable.
In terms of the quantity of the sample, do you need, when you're sampling of water, do you need less material than if you were to take a soil sample?
With the water, it's pumped through a filter.
Yeah.
And with a soil sample, you can't just pump it through a filter.
No.
So there's more complicated methods to get the DNA out. I've only done this once, and I'm not an expert, but we put you in touch with people who are.
That said, the filter that the material was collected on was about yay big.
Okay.
But only about a square centimetre of that was taken for each of the samples that we used to produce the DNA for these millions of reads. So the amount of material that you actually need is remarkably low.
You don't need an excavator.
We're interested in what species grew before they were native species grew before they were destroyed, particularly in the Auckland area. And it's been suggested that looking at pollen samples would be a way to do that. But maybe DNA replace that.
I'm not, as I say, I'm not an expert. We can put you in touch with you. I would have thought that there's some historical samples, in particular at Auckland Museum, that may well go back at some time. I've been a little bit involved with some of that, and there are even historical soil samples from Auckland Museum that could perhaps help.
Can we imagine a future where we have some kind of device stuck in the forest and it rings an alarm bell like a ferret or a stoat walks past? And if so, how close that stoat or the ferret have to be to trigger?
I don't think we give off that much volatile DNA, but certainly if you were monitoring the headwaters of a stream, there are, so ESR and Miles Benton at ESR, the scientist who's developed these tiny little computers that do everything this does, but he's automating this to go into streams and places around. And you could certainly, with telemetry, that'll transmit either through a satellite or through Wi-Fi, depending on geography, to a central place and give you a warning, or give you a real-time readout of the ups and downs of stoat or ferret DNA.
So if a stoat or a ferret plunges into a stream, then we could potentially capture it.
Yeah, especially if it goes to the toilet in the stream. That's, I understand, the best thing to do. As I say, we're not the experts, but we can put you in touch with people who are, if there's particular things you'd like to do. Please get in touch if there's things that we can do further with this. And I think I need to formally thank again Thierry and Annabelle and Rosemary and Scott for being part of this, Rowan for helping with the setup at the start, and all of Kim Handley's team. Kim runs a big science team in this area at the university to help with sampling. And in particular, the school students you saw there and their teachers who were a big part of the project. So thank you very much. And I think the next session starts at 1:15.
Yes, in just a few minutes downstairs. You'll see a notice on the door.